Hello, everybody. In this lecture, we're going to talk about regulating protein synthesis. So let's get right into it. Now, by the end of this module, you should be able to describe the difference between constitutive and facultative genes, summarize the effects of pre-transcriptional regulation, and the basic role of transcription factors in regulation, define the parts of an operon, and compare and contrast inducible and repressible operons, explain what epigenetics are and how it impacts protein synthesis, describe quorum sensing and its role in protein synthesis, and define post-transcriptional regulation and provide specific examples of it. All right, so controlling protein synthesis is essential for all cells. Protein synthesis is going to be regulated through both pre and post transcriptional methods. It is going to use, like when we're making actual proteins, it uses a lot of resources and a lot of energy. So if you're making a protein that has 220 amino acids, for example, you are of course using all of those amino acids. You've got to synthesize all the NTPs for your RNA. You're holding up all of those individual tRNAs that need to bring amino acids into the ribosome. You're using those ribosomes, etc. And on top of all that, there is a significant RNA, energy requirement. So to charge a tRNA, so charging the tRNA is the process of adding a amino acids to a tRNA. The charging process takes approximately 2 ATP. And then when you add a new amino acid to your protein chain, that takes another 2 to 3 ATP. So on average, making each individual amino acid in the protein chain is going to cost somewhere between 4 and 5 ATP. And so that energy could be used somewhere else. It's very important for the cell to be able to control this and only use those resources and that energy when it needs to. And so we do this primarily through this process of having both housekeeping and facultative genes. So not all the genes are going to be constantly expressed at all times. In general, only about 20% of a cell's genes are being expressed at any given time. And our housekeeping genes are genes that encode proteins that are going to be found in the cell and are considered constitutive. Constitutive genes or housekeeping genes are basically always turned on. They are required for life. If they are turned off, the cell cannot function. Facultative genes, on the other hand, encode proteins that are made in response to a specific environmental change or a special job. And so if you think about something like the term facultative, which we've talked a little bit about in class, in the sense of like a facultative anaerobe, that facultative anaerobe likes to live in an aerobic environment. However, if it is in an anaerobic environment, it can do so by facultatively turning on the genes that allow it to live in that anaerobic environment. And so we have several different pre and post transcriptional regulation pathways. And we're going to talk about these both. We're going to talk about both of these in just a moment. We're going to talk about them primarily in a general fashion, as there is a fair amount of overlap between eukaryotic and prokaryotic mechanisms, although there are some specific differences, and we won't go too deep into those. And when we think about our pre-transcriptional regulation, we've got a couple in particular that we want to talk about: transcription factors, operons, epigenetics, and quorum sensing. And these are all going to largely be driven by changes in the environment that allow them to like take and sense those in changes and then make alterations to what they are making in the cell. So we start off with transcription factors. These are types of proteins, and they help turn specific genes on or off by binding to the DNA nearby. It's kind of demonstrated in this picture on the right-hand side. RNA polymerase doesn't bind super well to the promoter by itself, so you need these transcription factors to actually be able to get a good tight binding to the promoter and not get knocked off. We have activators, and these are transcription factors that are going to increase the gene's transcription. We have repressors, which are transcription factors that are going to decrease the gene's transcription. And then we have sites on the DNA where these activators and repressors bind. Those are called enhancers, which bind multiple activators, and then silencers, which bind multiple repressors. All right, so transcription factors, as I mentioned, are these proteins that help turn genes on or off. The activators can boost transcription while repressors decrease it. And then enhancers are sites that bind activators, and silencers are sites that bind repressors. So let's talk a little bit more about the activators. So activators are going to bind to like an enhancer site. These sites are going to be upstream of your promoter, and they are going to help promote gene expression. And so if we look at this little cartoon down here, we have the promoter downstream, we have an enhancer site here, we have two activators binding to that enhancer site. It's ultimately going to increase transcription. Here's kind of an example of something like that. So this enhancer site here is going to bind these three activators, which help bind all of this giant protein, all this big protein complex that's bound to your RNA polymerase. Whoops. And that RNA, and this basically holds everything in line so RNA polymerase can attach to the promoter site. And then once it's attached, it can actually proceed with transcription. On the other hand, we have sites that can be called, or that are called silencers, that can bind to repressors, and that is going to prevent the transcription of the upstream gene sequence, or the downstream gene sequence. And so that can happen in a couple of different ways. One, we can directly block the promoter, right? So some repressors can bind straight on to the theta box. That's that -10 region in the promoter. Other situations, like in B here, we have that the repressor actually bound upstream, and then it caused this hairpin to form where the repressor binds to the upstream site and then blocks the -10 region of that promoter. And then we have situations where the repressor, which is represented by the R's here, bind onto these activators, right? So it prevents the activators from doing their job. And I'm not expecting you to memorize these different options per se. I don't need you to be able to tell me all the different things that are going on. But if I were to ask, if I were to present you with a situation where I explain one of these to you, I'd want you to be able to identify it as being a repressor. Or alternatively, if I explained one of the previous options to you, being able to identify it as an activator. All right. So where transcription factors can be really important, especially in prokaryotes or in operons. Operons are a collection of genes that are controlled by a single regulatory element. They have four key parts: the promoter, the genes, the repressor, and the operator. So the promoter is the same thing that you would have for any sort of replication or any sort of transcription, rather. For the genes, we're going to have at least two or more genes, and they're going to be usually included in a shared task. And then the repressor is going to be the transcription factor that blocks transcription, and the operator is the place where the repressor binds and blocks the transcription. There are two main classes of operons. We have inducible operons and repressible operons. So by default, the inducible operons are turned off, and thus they can be induced to be turned on. And by default, the repressible operons are turned on, and thus they can be repressed to be turned off. So probably the most famous inducible operon is the lac operon or lactose operon. It controls the transcription of 3 structural genes that help the cell metabolize lactose. And the lac operon is induced only when lactose is present and glucose is absent. In that situation where lactose is present and glucose is absent, the cell makes an inducer called allolactose, and the inducer inactivates the repressor, allowing RNA polymerase to transcribe the structural genes. So here is the default state. In the default state, the repressor is going to be made. That's going to be a protein such as this one here. And that protein is going to bind to the operator. And when it's in the way of the operator, of course, is downstream of the RNA polymerase, so it's in the direction that RNA polymerase needs to go. And therefore, it's going to block RNA polymerase from proceeding across those lactose utilization genes. And therefore, transcription does not occur. Now, in the event where lactose is present and glucose is absent, the repressor is made just as before. But in the presence of lactose, you get the formation of allolactose, which is represented by this green guy here. Allolactose can bind to the repressor, and you see that it changes the shape of that repressor, preventing it from being able to bind to that operator. And since the repressor can't bind to the operator, transcription can occur. RNA polymerase binds to the promoter and goes on ahead. So that's what those two scenarios look like side by side. I highly suggest taking some time to understand that. Now, an example of a repressible operon, here we have the arginine operon or ARG operon. It regulates the production of the amino acid arginine. The ARG operon is transcribed unless arginine is available. And so when arginine is available, arginine binds the repressor. Repressor gains the ability to bind to the operator and blocks transcription. All right, so in the normal default state, the operon is on. All right, and so in this case, the repressor is made, but the repressor is inactive. It's not able to bind to the operator. And so as long as arginine is in low quantity, then the repressor is not going to become active, and the operator is not going to get blocked. And therefore, RNA polymerase will proceed through transcription, and it will make the transcripts that need to go to the ribosome to synthesize arginine. Now, in the inactive or in the situation where the operon is turned off, then you still get the formation of the repressor. However, arginine is in abundance. Maybe this is because the cell has made enough, or maybe this is because the cell is in an environment where there is a bunch of available arginine. But regardless, that available arginine is able to bind to the repressor, thus activating it to where it can now bind to the operator. And when it's bound to the operator, it blocks the progression of RNA polymerase, and transcription does not occur. That's what those two look like side by side. I would review this as well, kind of understand the difference between inducible and repressible operants. All right, so then we have epigenetics. Epigenome or epigenetics is the study. So epigenetics is the study of chemical changes to the genome, while the epigenome is the collection of all those changes. A major one is DNA methylation. So that's pictured here on the right-hand side by all these methyl groups. Those methyl groups are going to be bound to your DNA and they prevent transcription. So they silence genes. Basically, they make it difficult for the process of transcription to occur. Now, another thing that bacteria use is something called quorum sensing. This is essentially a type of cell-to-cell communication, and it what happens is the cells produce these communication molecules that are auto-inducible. And in low abundance, when they're just by themselves producing these, then they don't identify enough local, like they don't identify enough of the autoinducer to be available to produce the enzyme, and thus they don't. But when there's a big group of bacteria together, there's enough autoinducer molecules being produced that they can make whatever enzymes they need for that situation. Common time that this is used is during the formation of biofilms, which makes sense since biofilms are formations of groups of organisms. And here we kind of graphically see this or visually see this, where in this low densities environment, the autoinducer, which is this acyl homoserine lactone, is being produced, but it's not really in a high enough production for them to detect it. When they get a bunch together, then a bunch of autoinducers are being produced. And so then you can start to form that biofilm. So if we look down here to see what's actually happening, the autoinducer is being produced and it's exiting into the membrane. And then when it diffuses, it's going to find maybe the same exact cell, maybe a different cell. It's going to go into the cell where it's going to bind to these regulatory proteins. And that when they bind to the regulatory proteins, that's going to cause them to basically become active transcription factors. They can now bind the DNA and alter the transcription of different genes. All right. So we're going to talk briefly about post-transcriptional modifications. This includes altering mRNA stability, mRNA splicing and export, and small non-coding RNAs and ribo switches. And so there are several agents that can affect mRNA stability, both positively or negatively, right? So in some cases, you can produce things onto your mRNA that will block these enzymes that chew up the mRNA. Right. So up here is an example, this N6 2' O dimethyl adenosine. I do not need you to know that name. But what it does is when it's on the mRNA, it prevents these RNases, which are enzymes that like chew up the RNA, from chewing it up. And so that means that the stability of the mRNA goes up. On the other hand, you have something like this 8-oxo-G. Again, do not need you to memorize it, but it provides a place for enzyme complexes to basically come in, chop it up into smaller pieces, and therefore it reduces mRNA stability. So anything that is going to affect the stability of mRNA is going to be post-transcriptional because obviously that happened after transcription occurred. We also have small non-coding RNAs. These can come in the form of microRNAs and short interferon RNAs in eukaryotes or just small RNAs in prokaryotes. And what they do is they limit protein synthesis by binding to the mRNA. And so you'll have, in this example over here, we have two options. We have this small RNA, which is represented by the green, and it binds to the mRNA, right, which is represented by the blue. And if that mRNA is bound to this small RNA, then it can't get into the ribosome. Now, the cell can produce anti-small RNAs, which are basically sequences that will just bind up the small RNAs. And as long as they bind them up, then the mRNA can make it into the ribosome. And yeah, if the small RNAs are bound to the mRNA, then that tags that mRNA as being something that's ready to be degraded. And then last but not least, we have riboswitches. They're mRNAs that have these built-in coding sequences that act as switches that control protein synthesis. They contain some sort of sensor region, and that will bind a metabolite like a vitamin or a derivative of a vitamin. And here we have these three sections of a riboswitch. We have the 5' untranslated region, which is going to have the riboswitch in it. We've got the coding region, which starts at the start codon. And then we have the 3' untranslated region, which is going to be after the stop codon. So the riboswitch is going to be in this 5' UTR area. And what we can see in this image is on the left-hand side, we have a riboswitch that is not in the presence of its metabolite cofactor. And in this case, the translation, it can occur, no problem. However, when the metabolite comes into contact with the riboswitch, then you get this change in the shape, basically, which causes part of the sequence to be moved into this, what's called the aptamer domain. The important part here is when the metabolite is present, it prevents translation from occurring. All right. So with all of that, you should now be able to describe these differences between constitutive and facultative genes, summarize pre-transcriptional regulation, define the parts of an operon, and compare and contrast inducible and repressible operons, explain epigenetics, quorum sensing, and post-transcriptional regulation. Hope you enjoyed, and I'll talk to you soon.